medical school at Louisville in Kentucky, and then went on to do his general surgery. Not general surgery, did an internship in general surgery at Iowa, and then his ENT residency at Iowa. He then went on to do his fellowship in microvascular reconstruction for head and neck surgery at University of South Florida. You joined us in 2009? Yep. 2009, along with, back then, many of you may recall Dr. John Lee. While Dr. Spanos was doing his residency training, he was also very much involved in an NIH T32 funded research on HPV, and as many of you remember, that was Dr. Lee's very strong suit, and engaged Dr. Spanos, and I think a good reason that you came here and joined him to continue on with that research. Dr. Spanos has millions of dollars of grant money for which he's looked at a number of things. Currently, I believe he's looking at the mechanisms of radiation-mediated clearance for HPV-positive head and neck cancer. He's done research for radiation fibrosis to the head and neck. He's done research and secured grants for circulating tumors in head and neck cancer, as well as a $1.4 million research project on cisplatin-mediated immune modulation for HPV-positive head and neck cancer. What I'm telling you is he's doing real-time research here in South Dakota. Let me repeat that. He's getting millions of dollars for research here in South Dakota. And the whole premise of what he's going to be talking to us about this morning is you too can be doing that wherever you are if you're so inclined, and he's going to be giving us some tips to that. In addition to his research, which is heavy, he is currently an assistant professor, likely to change with the university's academic assistant professor, likely to change, we're believing, this spring to associate professor. He is currently your South Dakota State Chapter College of Surgeons president for the ACS. And in addition, that enables him to share a lot of his research experience with all of the surgeons in North and South Dakota. It's very much a trailblazer to prove or disprove the theory that, well, I'm too busy in private practice to do research. This is an individual who disproves that concept every day. This morning, Dr. Spanos will be enlightening us as he discusses translational research for surgeons, something he knows quite a great deal about. So please join me in welcoming Dr. Chad Spanos.
All right, it's my pleasure to talk to you guys. Hopefully we won't see too many people sleeping, so don't worry. I've pulled out a lot of the basic science slides, and I just tried to give you an overview of what translational research means. And this is not for everyone, but I'm going to try to make the pitch to you that you can play a role in no matter what job you do, even if you don't want to stay in academic medicine. So what is research, right? So, and as we go through, yeah, we may want to drop the lights a little bit because I do have some color figures. So, you know, when we think about research, I think especially back to undergrad and med school, this is probably what comes to mind, right? People going cross-eyed, staring through a microscope, pipetting things that look like water into small dishes, and you know, having faith that whatever comes out of the machine on the back end tells us something. Or even, you know, drug design, right? You're looking at organic chemistry and trying to design the next blockbuster drug, right? Or if it's been a really long time since organic chemistry lab, maybe this is the closest that you've been to research, which is watching CSI and seeing all the black lights and the computer that beeps and says positive, right? Well, I hate to say it, but I've never worked in a lab that had such cool lighting, and scientists definitely don't look that attractive. So when you want to fundraise, this is kind of how you pitch science, right? So this is from University of Kansas. And when you want to fundraise, you basically try to say, cancer is really bad, or some disease is really bad, and if we just invest a lot of money in research, then we'll bring some cures and we'll make everybody better. But we know it's all more complicated than that, right? Because if it was this easy, there'd be a lot of diseases that were cured out there, and there aren't. So this is idealism. I stole this from Duke's surgery department, which I think is actually pretty good place to start, right? So, and you can be involved in anywhere on the wheel here, right? So discovery, where the microscope is, those are probably more the PhDs and the undergrad students and the graduate students in the lab looking at disease mechanism, right? So working with cell culture models, working with things like that, trying to find, or even way back where looking at yeast and bacteria and trying to design new drugs and such. And then once you have something that looks pretty promising, that's where you can take it into this area called translation, right? So you're working with animal models, you might be working with some early phase humans, trying to figure out, is this real, right? Because the question is always, is what you observed in the Petri dish real, or is it just only going to happen in the Petri dish and it'll never happen in real life? And then, once that looks promising, what do you do next, right? You take it to the patients, because unless it actually works in patients, we don't care about the green area, we don't care about the microscope, right? Unless it actually works in humans, unless it actually cures a disease or improves a disease or alleviates suffering, it's not helpful. And that's where the testing and the clinical trials occur in the application phase. I would add another little section in here, and I hope to highlight a project that shows why this section is important. There's another section in the orange category, you can see my pointer there, it should be right about here. And that's actually specimen collection, observation of interesting findings that you have in your patients. So just like what you found in M&M, if you notice a pattern, if you say, every time we do this, or here's a major problem that we have and we can't figure out why this is happening, those kinds of observations of patients in the clinical world, and especially if you have specimens, can then lead to more discovery, right? So that's where you go back and you partner with the scientists or the PhDs to try to understand what's happening, and you keep the wheel going, try to come up with new treatments. So the realism, not to throw cold water on Duke, I don't like their basketball team anyway, but the realism is that this is actually a very long, very huge process. So you can see over here that you have these laboratory tests like we talked about, you got to write a research protocol, you have to make PETA happy, you have to do all this animal testing, or they would prefer you do none. And then you go through clinical trials. And at the bottom of the screen here, you're seeing that more and more and more patients are required as you step through the phases of a clinical trial, right? So just to remind you, phase 1 clinical trial is safety, asking the question, is this safe? Phase 2 clinical trial says, not only is it safe, but are we seeing some type of efficacy symbol? And then phase 3 is comparing to the standard of care and saying, is this any better or is this any worse? And that requires sometimes thousands of patients, significant cost. And at each step, always the risk that you're going to come up with a big goose egg and say, this didn't work. And then your drug gets approved, and even after drug approval or after new therapy approval, so this could apply to devices as well, you have to follow and say, does this actually cause any unexpected complications that we didn't see in the clinical trial testing, right? So it's a long road to get something that is accepted, safe, and that's what this right-hand side is showing here, right? So the big stacks of dollars are coming up, especially as you get into phase 3. And so if you ever wonder why something takes so much money to get the clinical efficacy, this is why, right? So they say, worst case scenario, 15 years and over $1 billion just for one single drug to get it through all those hoops. So it's a big trouble. And if you look at this left side of the screen, how much did it cost Da Vinci to do all the robotic testing? And this probably started way back when in MIT with some of the engineering labs, and then to take it from a concept to something that actually would be safe for use in humans, to do those early clinical trials and then have it approved so that you could actually do robotic surgery safely. That's probably a big price tag. Is it as much as Da Vinci charges? Who knows, right? Unless you look at their books, we don't know. If you look over in my world, which is the oncology medicines, especially the immune therapy medicines, the cost is significant on what we're spending on these drugs, right? And the drug companies would say this is because of all of those input costs that you saw on the previous slide. Subject to controversy. So I can't talk to you about general surgery as much, but I will try to tell you about is a couple case examples of translational research in the head and neck world. And as we go along through here, maybe you can see little areas where perhaps this would make sense for you, right? So, well, I'm not going to run a research lab, I'm not going to work with animals, but I can do this. Or, I really don't enjoy doing observational studies, but yeah, I'd like to participate in a clinical trial. And participating in a clinical trial is sometimes as limited as saying, I do surgery on breast cancer patients. I'm willing to sign them up for a trial, to talk to them about the trial, and work with the local investigators to help answer this question. It really can be as complicated or uncomplicated as you want. So head and neck cancer, if you don't know, is the 6th most common type of head and neck cancer.
Attention, adult level 1, trauma team to the emergency department, room R2. Attention, adult level 1, trauma team.
So human papillomavirus causes many of these cancers. It's very toxic therapy for chemotherapy, and radiation surgery also plays a role. There's a lot of room for improvement. That's the summary of this slide, is that head and neck cancer stinks and the current treatment's not great. So without going into all the details, what we did when we were back at Iowa, this is John and I, is we decided we wanted to figure out a model system for head and neck cancer. So we developed a tumor system in a mouse where it had one of the most common causes of head and neck cancer, which is human papillomavirus, actually in the tumor. And then we put these tumors into mice, and we did some of our conventional treatment, right? So we treated them with radiation, we treated them with chemotherapy. And in the mouse, you can do all kinds of artificial manipulation that you could never do in humans, right? In the mouse, we could say, well, what happens? What happens if we have an immune system? And what happens if we take away that adaptive immune system? And lo and behold, it was very interesting that when you have your adaptive immune system in place, if you use high enough doses of radiation, tumor volume is actually low and cured. When you do not have an adaptive immune system, that's over on the right-hand side of the screen, even at high doses of radiation, you cannot cure any of those tumors. And that was a big aha moment where we said, immune system is playing a huge role. And that led to some of the other things I'm going to show you today. So a lot of this work is not done by me. Whenever you see any research that's done, and whenever you have an invited speaker get up and say, this is everything that I did, you can call them out on it, because what it really is a bunch of people working with that person. It takes a team to do a lot of these research projects. So this is Dohan. He's one of our collaborators over at the University of Colorado, and the next few slides are a lot of his work. So basically, we're gonna talk about something that Dohan found within clinical specimens, right? You don't need to remember the whole mechanism. That's not why we're here today, is to learn something new about HPV. What I want you to watch through these slides is, what did Dohan actually need to figure this story out? And what he needed was actually samples from cervical cancer specimens and from human head and neck cancer specimens to be able to find this question out, right? And so we're going to talk about CXCL14, which is an immune cytokine. So it's essentially something secreted by cells that had been known previously to cause macrophages and T cells to infiltrate into a wound environment. So what do you do, right? When you can't figure something out, when you can't figure a disease out, you go back and you say, what's the natural history of the disease? And so when we look at HPV progression, especially when we look at cervical cancer, we know that there's a continuum, right? So you have normal cervix, and then you get a low-grade lesion, then you get a high-grade lesion, then you get cancer. And as those things progress, you get many different changes within the cell, right? So there's a lot of genes that change. And so what he did, which was really smart, is he compared those gene changes. Again, these are human specimens, compared those gene changes and the progression from no cancer to high cancer. And he found one thing, that as you go from no cancer to cancer, it went down significantly. Wonder what that thing is, and why is that important? So you can't just go straight into clinical trial and say, well, we'll restore that to the cancer cells and everybody will be cured, right? If it was that easy, then this would have been done 5 years ago. But you have to figure out actually what it's doing, so that you know what it's doing, so that you can design the therapy so that you don't have unintended side effects. So this is again just showing that in cancer there was a lower level of expression of this CXCL14. And then we wanted to say, does it actually cause cancer? So you have to go into cell culture. So these are actually cells growing in cell culture, and when we have cells where we delete this HPV gene, so HPV has different genes that it uses to cause changes in the cell. When we delete those HPV genes, look at this, CXCL14 actually comes back up. So we knew that it was due to HPV in these cells, why we were seeing that decrease in CXCL14. So same thing, this is another cell culture model. We looked at cervical cancer cells, and as you have a progression from normal cells to these other cells, you can see a change in E7 expression. You also see a corresponding decrease in CXCL14 expression, suggesting again that this is HPV causing this, right? So we're getting more information to say that HPV is doing this. So we've taken information from the clinic, we've now adapted into cell culture models. We're trying to understand more about what this thing is doing. And lo and behold, we knew that one of the things that this HPV oncogene does is it goes in and it modifies your genome. So has anybody heard of epigenetics? No? Okay. So essentially this is a way for you to turn on and turn off your genes, and it can be a way that actually is inherited. So you basically, remember your DNA is wound up into these little balls, chromatin, remember that all thing? Okay. So when it's heavily methylated, it's really, really bound tightly, and you can't get read through of genes. And when it's not methylated, it tends to open up more, and those genes are read through, and those products are made. And it's a way for the cell to control what's happening. And so the virus comes in here and it says, yeah, we really don't want this CXCL14 around. We don't want the immune system coming in and giving surveillance to the tumor. Let's just shut that area down. So it heavily methylates that area, and then you don't get expression of CXCL14. And that's essentially what we found that HPV E7 was doing. So does this actually matter, right? So if you remember that slide way back when, we were talking about in the Petri dish, looking under the microscope, doing our fancy genomic tests. But does this really matter for tumor growth, right? Well, tested in the mouse. So now we're looking at different mouse cells. We've now engineered to restore the CXCL14 into some of the mouse cells. We put them into mice for tumors. And lo and behold, if you don't have the restored CXCL14 in the tumor, all the tumors grow out and the mice die, right? So this would just be like uncontrolled, untreated tumor growth. When you restore CXCL14 into those tumors, some of them clear, which is kind of amazing, right? For cancer to be cleared by the immune system alone, but some of them actually will clear without any treatment just by restoring CXCL14. So we're pretty happy about that. And does this affect the immune response? And so I don't want to get into this too much, but we basically found that this was a result of the adaptive immune response. So again, these are these same mice that don't have B and T cells. When you put the tumors into those mice, none of the tumors clear. So we knew that this was an immune-mediated mechanism. This is just talking about NK cells and MPSCs. We don't need to go into that. So that's one story. So where is that project, right? So that's really cool. We've now cured some mice. Yay. Do patients care about that? No. And so what's happening next is that data is encouraging, and it's maybe going to make it into a therapeutic product. All of that depends on venture capitalists, whether they think that this story is compelling enough. Do they want to invest their hard-earned money into the next phase of that uphill battle? And sadly, this really comes down to economics, right? So is the potential return worth the risk of investment? So even though you have a really great, compelling scientific story, it may never make the light of day if the financial return is not enough, or if someone doesn't think that this is a good bet. And so stay tuned. We'll see if that ever goes anywhere, but we're hopeful. So this is a story that's more mature, right? So now I'm going to show you a story of something that actually has made it into clinic. And these are patients that you probably see when you're on your oncology or your especially your breast rotation. Some of the breast cancer patients are now receiving this immune therapy, and the lung cancer patients definitely receive some of this. So this has been a revolution in oncology. So up till about 10 years ago, what we had for immune modulation was basically IL-2. which is interleukin, right? And it's very toxic, doesn't tend to work too well. And there was this one researcher down in Texas, I can't remember which institution he was at, but everyone thought he was insane. They wouldn't give him any money, but he kept plugging along because he knew he was right. And he's the guy that actually found a different molecule on the screen here. So I'm showing you PD-L1. He found CTLA4, right? And he developed essentially the drug now that's used to block the interaction of the T cell and the tumor cell and actually turn on the T cell so that it kills cancer. And before he proved this, everyone thought he was insane. Now he looks like a genius, right? So it just shows you how common opinion is not actually always correct in science, and you can have contrarian views. So I bring that all out because if you're sitting there and you are working in a smaller practice and you're noticing something, you're noticing a pattern that goes against what the big guys at the university say, right? You might be right. Check your data, follow those patients, do some tracking of outcomes, because you might be right. Just because the big guys say that something is the way it is doesn't always mean that that's correct. So the story that we're talking about here, to bring this back to this example, is tumor cell versus immune cell, right? And we know that for tumor cells, most of the killers for the immune cell are these activated T cells, right? The CD8 positive T cells. So we have them floating around in our body. They get into the tumor microenvironment. We know they're there. Why does anyone get cancer? This was a mystery. Well, what they figured out was there's actually all of these molecules sitting on the T cell and sitting on the tumor cell that interact. And when those molecules interact, you know what it says? It says, I'm your buddy. You don't need to watch out for me. Don't kill me. I'm fine. And that's actually why the body is tolerant to cancers in many situations. So here's a little schematic of how this works, right? So the tumor has PD-L1, the T cell has PD-1, they bind, boom, tumor is going to be destroyed. But, if they don't bind, if they don't bind, tumor is going to be destroyed. If they bind, then that tumor is labeled as friend, and the T cell goes away, or the T cell gets turned into what's called a T regulatory cell or myeloid-derived suppressive cell. So it creates this environment that prevents autoimmunity. Okay, this is the reason why when you get an infection, you don't lose your entire arm because the body goes in to try to destroy everything. It stops once it realizes that it's friend or foe. So for head and neck cancer, here's an example of the PD-1 inhibitor, or this treatment, had been already taken through the lab, discovery, through animal models, and then it was now produced by a pharmacology company, and they were in phase 1 trial, right? So does this even work? And some of the first patients that they actually tested this on were head and neck cancer patients. Do you guys know why? Because you can actually see the tumor, and you can watch response, right? And it's not in a critical organ like the lung or the liver, so that toxicity should be somewhat manageable just in case you get a massive immune response. So here's a guy that's been through, you can see he's been through radiation, he's been through chemo, he still has tumor infiltrating all throughout the skin. This is dermal involvement. And 1 month after, you might start to think, uh-oh, this really isn't working, we're in trouble here, but actually, this is tumor flare, right? This is such a massive, robust immune response that it's red and it's swollen, but then within 3 months, the lung mets are gone, this is starting to look better, and by 6 months, it's a near complete response, right? So it's pretty amazing. And this is a breast cancer patient, so just so you don't think that this is all head and neck, this breast cancer patient, that's pretty bad, right? I'd say I'm not a general surgeon, but that doesn't look good. And with a single treatment of immune checkpoint inhibitor, cancer is essentially gone. Now you've got a wound that you have to deal with. Call Dr. Wynn, right? So, so this is another cancer patient, lung cancer, stage 4. It's already gone through multiple lines of chemo, 4 treatments with PD-1 inhibitor. Now you're getting a response. And that's the truth in advertising, is that there's some people that this works for, and there's a lot of people that this doesn't work for. So if you look at head and neck, about 50% of people had a decreased tumor burden. The number of people though who had a complete response, you're looking more at maybe at best 10 to 15%. Melanoma, that's actually really impressive. Melanoma has been a rock star for this immune therapy, and you get a significant response. So when we saw that data, right, so that's early phase 1 data looking at recurrent metastatic head and neck cancer, we thought, well, and this is a different type of translation, right? We thought, well, can we bring this back into treatment of patients who have never seen a different therapy before? So can you take that drug and can you go straight into somebody who has never had cancer treatment? Well, yeah, now that we know that these drugs are safe and we've had a lot of experience with them, you might be able to convince an IRB to do that. At this point in time, that was a risky proposition, right? That was like, whoa, what are you guys doing? So we went back to the lab to try to gain some efficacy data to say that this is a rational approach. So we used this anti-PD-1 inhibitor in our mice with tumor growth. It doesn't respond to anti-PD-1 inhibitor alone. When you add chemo and radiation, which is our standard treatment for head and neck cancer, you did see an improvement. This is this lower line compared to the upper group that just had chemo radiation, and we saw about a 50% survival spread. So that was really encouraging, right? So we said, okay, let's do this. And so basically, there's other approaches that we can use. And so we basically thought about stimulating this with a virus. So synergizing between the immune therapy plus a virus, the virus would actually prime the immune system to recognize the HPV in the tumor cells, and then that would really create a firestorm of an immune response to the cancer. And so we basically developed this adenovirus. You guys know adenovirus, right? Cold, you get it every year. You can actually grow those in the lab, and you can put whatever genes you want into the adenovirus. So it's kind of like the Trojan horse, right? So you could throw in HPV genes, you can throw in other types of cancer virus oncogenes, whatever you want in there. And you can essentially vaccinate someone with those viruses and hopefully stimulate an immune response to that payload. So what that does, just like when you get your series of 3 hepatitis B shots, you're now boosting your immune response up to a high level so that when you apply treatment, the immune system says, aha, I know this guy, this is not my friend, I'm really going to go in there and destroy it. And that's essentially what we're showing here. So we made the adenovirus, we have this idea that it's going to work. So we want to test it in the mice, right? So we basically want to say, does the virus work by itself? Nope. Does the virus work with the tumor alone? Yeah, it can. It's not great, maybe about 30% response. But when we combine the immune therapy plus the virus, we get a much better response. So, and I think this is going to be a common theme going forward with cancer care, is that there's not going to be that single blockbuster drug, right? There's not going to be the antibiotic that cures all infections. It's going to require combined therapy, and it's going to be different for different tumors. And so this is just looking at our combination therapy, virus plus anti-PD-1. That was the immune modulation plus the viral vaccination payload. That looked about the best. It showed improvement over the PD-1 alone, which also showed improvement over standard therapy alone. We don't have only 10% of our patients survive chemo radiation treatment. What you can do in the lab, which you can't model in clinic, is you can actually game the system to say, worst case scenario, really, really bad advanced cancer, huge tumors, let's treat with conventional therapy, and then let's see, can we make it better, right? So you're trying to show that there's improvement with your experimental therapy compared to not doing your experimental therapy. Why is that? Well, if we looked at this and we saw that PD-1 therapy really didn't make any difference compared to standard therapy, why would you keep going with this line of therapy, right? Why would you even take this into clinic? Or if we tested our virus and the virus didn't make any benefit compared to using the immune modulation alone, why push forward? Why take that into clinic? So this is why you do these animal experiments, to provide rationale, because you have to think about yourself as the investigator in the clinic, right? So remember when I said you guys could help by being that principal or that investigator that talks to your patient and says, you know, we have this trial. I'll throw a hypothetical out there with a device, right? So let's say you're Dr. Kelly. We have this trial. I've got this new stent. We're gonna try it in you if you're okay with that. But I've never used it before in an animal. I have no idea what it's gonna do. I haven't even worked with it in the lab. We're just gonna try it on you. Is that conversation gonna go very well? No. But if you have that conversation with the patient and you say, you know what, we've actually designed this device. It's gone through rigorous testing. I've seen the data. Even if you didn't do it yourself, I've seen the data. I believe in it. We have reason to think that this will help you. That's a conversation you can have with your patient to say, this is worth the risk. And maybe the patient will say, no, it's not worth the risk. But for the right patients, I think these clinical trials do make sense. And so we didn't just stop there, right? So fortunately, this was a drug that had been developed by that scientist from South Dakota or Texas, P.J. Allison, then had been produced initially by Merck. Now there's many other competitors out there with all the different oncology companies. And Merck was willing to pay for a clinical trial. So this is now this translational story where it's gone from petri dish, animal, and now I'm going to show you kind of what happened in humans. And so this study was adding the anti-PD-1 inhibitor, so this immune modulation, plus our standard of care chemo and radiation for treatment of advanced head and neck cancers. We don't have the virus story for you yet. The virus is another one like CXCL14 that it hasn't gone into production, may never go into production, but at least we have this that we were able to take forward. So you've seen this schematic on the previous slide. So this is just testing the question of if we block this interaction between PD-L1 and PD-1, between tumor cell and T cell, will that actually improve the recognition of the tumor by the immune system, improve clearance? Boy, that would have been fun to do that trial initially, right? But that's not what we could do, right? So remember your phases of clinical trials. So we couldn't go straight to asking the question, is this better? Is this going to cure patients, which is really what we care about? We had to start with, is this safe? So this is a phase 1 trial to just say, is this even safe? Can we do this? So we know that there's activity in recurrent metastatic head and neck cancer. We told you about that, but we didn't know would it be safe to do this and combine with conventional therapy. So the way the study works, you start with your immune therapy up front, you quickly add in radiation and chemotherapy, and then you continue the immune therapy after radiation for a period of time. And then you watch these patients, we get PET scans, we get studies. The main primary endpoints of this study, though, were not, did it work? It's actually, did it hurt anybody? So, yeah. So you can see the patient characteristics here. Stage 4, most of them are stage 4, so pretty advanced cancers. Almost all of them have pretty bulky nodes in their neck. They've got advanced tumors, so that means larger tumors than you would be able to cut out with minimal surgery. A decent number of smokers, that just reflects our population here. And head and neck cancer population, they're usually older or on the upper side of the age demographic there. So, HPV status, this is kind of where we live in the Midwest here. So when you have cancers in the oropharynx, so in the tonsil, base of tongue area, almost all of those are HPV positive. When you have cancers involving the larynx and hypopharynx, those are almost, there's a decent number of those are HPV negative, and the reason for that is those are more smoking related. And we still unfortunately have quite a few smokers around here. So, all right, so was it safe, right? So surprisingly, yes, we were very happy that we had 3 discontinuations of just the pembrolizumab. Everybody got their, I think everybody got their chemo and radiation, but yeah, just 3 doses of the pembrolizumab. Some weird side effects, some really weird stuff happens with these autoimmune drugs, right? So one person got a Lermite type syndrome, so that's where he kind of bent his neck forward. He had some radiating tingling down the spine, some numbness in his extremities. Another one had some weakness of a leg, so almost kind of like a transverse myelitis syndrome. Fortunately, those would go away with stopping the drug and with giving high doses steroids. let's see, I'll show you here. So yeah, compliance with chemo radiation was great. And so that gave us reassurance, right? Because if you're looking at the safety of this and you're saying, if we give this immune therapy and then people don't get their full dose standard of care treatment, well, that's not very helpful, right? So we wanted to make sure that they could at least make it through treatment, and they did. You know, your standard, this treatment is fairly toxic, so you're always going to have some hospitalizations, and so we had a few of those, but nothing significant there. So this again reflects some of the adverse events that we typically have with head and neck cancer treatment. So almost everybody gets a dry mouth. They get what's called mucositis, which is, I explain to patients, it's like getting a sunburn in your mouth and throat. It's really no fun. It unfortunately comes along with chemo and radiation. You're going to lose some neutrophils, you're going to become anemic, you'll have some electrolyte disturbances, and most of those are just grade 3, so they're not severe, but they're noticeable. So we had one grade 5 event. The base patient died after treatment due to a GI bleed. He had a suspected GI bleed. He didn't pursue intervention. We recommended he did. We don't think that it's related to the pembrolizumab, but we still report it anyway because if you remember back to that schematic of clinical trials trying to find adverse events and then follow-up even in the years after the study drug's approved, over time, and especially as you see your patients and you notice these things popping up, there have been new findings that popped up with drugs way after trials were done, right? So you guys remember the Celebrex story, right? That issue with cardiac toxicity was not found until the drug was actually approved and been out in use in the population for quite a while. And unless people actually notice these things in their patients, report them to the FDA, or track your own data or do your own little clinical projects, we won't know this, and people will be hurt. So it's important to follow these adverse events. So the overall response was about complete response we had in about 80%, partial response in 15%, and then that one death. And so fortunately, we had a previous clinical study that we had done looking at chemotherapy and radiation plus and minus a metabolic modulator. So it turned the tumor environment into a less acidic tumor environment. Why was that fortunate? Well, we had a great control group, so we could look back and we could say, compared to that group, did our intervention in this group, was it significantly worse? And no, it was not. These numbers for partial response and complete response were pretty similar to what we saw back with our other clinical trial. And so that, obviously we can make no comment on this is better, but I think we can at least say that we're not hurting our patients, and this is encouraging to go on to next phase clinical trial. And if you break it down by HPV positive and HPV negative, well, we already knew, and the entire field already knew, that HPV positive patients do really well. For whatever reason, their tumors respond to treatment quite well compared to the HPV negatives. And that held up in this area as well, that you saw 85% of the HPV positives had a complete response to treatment, whereas only about 60% of the HPV negatives did. And so, right now, the study's ongoing. The Merck added a few extra patients to try to get larger numbers so that we could actually have some statistical significance at the end. And so we're continuing to do the trial. The interesting part is that we're now going back to the lab, right? So we're keeping that circle going. So the trial was written so that not only would we give the patients investigational therapy and track outcomes, but we'd actually take samples from the patients. They would donate their blood during treatment. And so what we have is we have tumor that's going to be analyzed by John Lee out at his company for, they're going to do whole genome. They're going to do full RNA expression profiles throughout the tumors, and they're also going to do proteomics. So in case you guys don't keep up with the oncology world, what you can do from a technological standpoint on a paraffin-embedded specimen is absolutely amazing now. You can do whole genome sequencing, so that tells you what's every last little DNA base pair in that tumor. You can do full RNA expression, and this is not just targeted, this is complete RNA sequencing. So every last RNA expression profile in that tumor, you can get it. And you can do proteomics, right? So they can actually take the tumor. I still don't believe that this works, but it does. It's kind of almost like sci-fi, right? So you can take the tumor, you can dissect the tumor out on a microscope slide, blow it up, send it through a mass spectrometer, and they can give you about 300 proteins that they know of their structure. They can match the proteins in the tumor to about 300 known proteins, and they can tell you HER2 new high, HER2 new normal, ER high, ER normal. It's fairly amazing. And so they're going to do all that testing on these tumors. And why are they going to do that? Well, if we find some interesting signals from this, we go back into the lab, we test things in the lab, and this might actually drive new discovery for treatments. We're also going to look at the immune cells. So we've harvested T cells from the circulating blood, and we'll do flow cytometry on those T cells and see if there's any interesting immune signatures. So kind of completing that circle, trying to take things back into the lab, try to do discovery off of the clinical trial. So I'll probably give you a little bit of time for questions. So basically, whenever you hear the keyword translational research, I don't think it means anything. I think it just means helping patients, right? It's basically why we went into medicine in the first place, right? We wanted to help people. And unless we actually continue to try to improve things, we're not going to change how we care for or cure patients. And so each one of you is a key participant in research, and it doesn't have to be working in the lab. It doesn't have to have an NIH-funded lab. You don't even have to be at a university. A lot of good research is actually done nowadays by people in private practice participating in clinical trials. And so play nice. When I say play nice, that means look for those opportunities. If you have a scientist come to you and say, hey, you know, Dr. Tamby, I noticed you do a lot of hepatic surgery, or you get samples, you're down that area anyway. Do you think I could, you know, would it be safe? Do you think your patients would be willing to give me a little bit of this sample? Because it would really make a difference in this question that I have. You know, those are the projects that you can really help with. Not everybody has to work in a lab. You don't have to, you know, have goggles and pipette and be down in the trenches. Sample collection availability is very, very important to keep this loop going. And process improvement is actually important. So just keeping your eyes open, noticing trends in your patients and your outcomes, and asking questions, why is this happening? And if you don't know, there's a lot of people in translational research or in science who are happy to discuss with you. And your crazy idea actually may not be too crazy, and might actually lead somewhere. So, yeah. And with that, I'd like to take any questions.
Okay, so Chad, since you've been here, and I will let the residents know, we have known of your HPV breakthroughs and then watched appreciatively as you were able to work that and presented your data at our college meetings and the like. And simply sitting in the audience thinking about that, I've got to tell you that I do the same thing then and say, well, is there anything in my field or observations? And 5 or 6 years ago, I made the observations that squamous cell cancers in the esophagus were getting better cure rates with neoadjuvant than they were with adjuvant therapy. And then thinking the same as you, this is a population of smokers, drinkers, and then had one breakthrough patient who had an HPV malignancy of her throat, who then later went on to make a squamous cell cancer of the esophagus. And began to wonder myself, is there a relation of squamous cells? Are there two different subsets of squamous cells, malignancies in the esophagus? And so in our world that I do esophageal work, I'm actually excited when I see a squamous cell. They're becoming less frequent. But I know that those patients do remarkably better. And in fact, when we go to resect after their neoadjuvant, the cancer's gone.
Yep.
And that was never the case 25 years ago. What I guess is it's a question, but also a statement. One was, are they applying now your technology to the esophagus and those malignancies that are also squamous cell in the lung and the like? And then just also the observation that what you do here makes other surgeons think about their own cancers they treat, and could there be an affiliation?
Exactly, and I think that's exactly true. Originally, when we started this story, we said HPV, just very simple thinking. HPV, it's a virus, foreign to the body. Well, of course there's going to be a better immune response. And actually, strangely enough, that's not true, right? So what really matters, and what we figured out now, is what really matters is the number of tumor neoantigens. So when you have smoking and drinking, like with some of those other esophageal cancer cases or lung cancer cases, that drives a lot of mutations within the cell. Some of those mutations will actually be made into proteins that are completely misfolded. They're so different than your native proteins that when those are expressed in the cell, the immune system recognizes those as foreign, as non-self. And so when you get these tumors that are highly mutated, they actually can respond to these immune therapies or to even neoadjuvant chemotherapy very well. And that has been a big change recently. And so for lung, I would say for almost every solid tumor malignancy, if there are not already clinical trials thinking about it, there are clinical trials being planned thinking about neoadjuvant chemo, because and chemo, when we say chemo, sometimes that is good old carbotaxol, sometimes it is these immune therapies, right? So chemo, neoadjuvant treatment of something to try to prime the immune system to attack the tumor. And it seems to be working very well, because when you go in for surgery, again, this is all anecdotal, but in my experience, margin control is better. I don't have these little feelers of tumor cells heading out into the periphery. There's a nice defined boundary to the tumor. And we're also seeing, if not nodal regression, sometimes complete resolution of positive nodes.
I have patients with squamous cell that then refuse the esophagectomy that are still alive.
Yeah.
And it's amazing, the only thing they've gotten is their neoadjuvant treatment. I don't have many adenos that are that way, but only the squames.
Yep.
And like I said, thinking outside the box opens the opportunity for other research. Hey, the other question I had is, at the beginning of your discussion, you pointed out that XLA454443 high.
Oh, CXL14.
There you go,
Okay,
that one. So you have that particular marker. Have you done anything with that that's in your back burner and thrown it with the IL?
Yeah, so that one is, that project fortunately is funded by a multi-PI grant with the National Cancer Institute between Dilhan, who's at University of Colorado, and ourselves. So we're looking at that in the mice. But John also is trying to round up investment and capital to potentially develop a therapeutic product with that to take into clinical trial. But if it did, that would be, 5+ years down the road. It takes a while to get it there.
I think it looks promising.
Yeah, it's actually a really interesting product. So it was originally found in the gut, right? So it was actually found it's an antimicrobial peptide, and it's something that the basal layer of the mucosa all along the GI tract secretes, and it naturally, they don't know exactly the mechanism of how it works, but it's thought to go in and somehow inhibit or kill bacteria. And so it's a natural protective mechanism, right? So it would make sense that if you have a virus that wants to establish and replicate, that this would be an area that it would want to downregulate, because otherwise, it could do it. Yeah.
It's a great talk. Thanks, Jerry. I think maybe 3 or 4 of you are in the residency when Dr. John Lee took Grand Rounds. I think his take-home message was similar to what Dr. Spanos said. With cancer care, as surgeons, you need to drive that bus.
Yes.
You need to help lead that effort. If you don't, you're going to be left out. And he repeatedly says, I heard him say more than once, that I'm happier to be, as an oncologist surgeon, work myself out of job.
Yep.
That's a good day for the patients that we're taking care of. So surgery is not always the right thing for patients, for cancer patients. So I think, I reiterate what Dr. Spanos said, when you go out in the practice, be engaged and work as a team, otherwise you're going to be left out if you want to stay. And even ask Dr. Timmerman, who graduated about 50 years ago. I mean, did he ever think of doing laparoscopic surgery? Did he ever think of robotic surgery? Trust me, in 10 to 20 years from now, when you're in practice, you'll be doing things that you didn't learn here. So keep that open mind, and only way you're going to have a job is to play in the sandbox with others.
Yep.
That's huge.
I'm going to even expand on that. 30, 40 years ago, head and neck cancer was a death sentence. They had horrible deaths. Even after the operations, it was abysmal. What would you say the percentage of surgery has been lost since the development of the chemotherapies, radiation therapies, and now your immunologic therapies?
If you look at ACGME, this is more germane to the residents, but if you look at the ENT-related case numbers that are required to graduate, residents are now, if you compare it to like 20 years ago, residents are now not getting an adequate number of laryngectomy cases of some of these really advanced morbid resections, because those advanced stage cancers are getting what I showed you in the talk of the chemotherapy and radiation combined therapy, and that actually works quite well in many situations. So it has been an evolution. It's not all doom and gloom though. So I know John and I would always say, well, we're working to put ourselves out of a job, but I think the story is evolving a little bit, and there is some hint in the neoadjuvant chemo or neoadjuvant immunotherapy world that if you prime the immune system to debulk the tumor, to take it down to a small size, and then you chase it with surgery that hopefully would be minimally invasive, minimal morbidity, but you remove that chunk of tumor that's like an abscess, right? It's a sink for all the immune cells to just sit in there and fester. There is some hint that actually might be a really great way to go. So stay tuned. You might not be out of a job yet, but I think that yes, I mean, we should all work for our patients, even if that is at the detriment of our checkbook and our career, right? I mean, it's kind of, again, if you think back to when you were ideal, when you were in college applying to med school, that's why we all went into practice, not to have a big house and big cars. So go ahead.
So how much of your practice, or I guess how much do you spend, like how much does research impact your like personal life, as far as do you have to spend a lot of nights here? You know, like how much time do you spend of your clinical practice in the lab, you know?
Yeah, no, I totally understand what you're saying. Yeah, so I had to negotiate this up front. So if this is something that you want to do to the level that I'm trying to do it, and I won't pretend to say that I'm doing it really well compared to people elsewhere, but you have to negotiate that up front. So you have to find a position where you say, I need 1 day, I need 2 days, whatever protected time where I cannot be scheduled in the clinic, I cannot be scheduled in the OR. You have to negotiate that up front. So I spend 1 to 2 days per week doing research-related activities. That is the other caveat to this, that you have to enjoy doing it and you have to want to do it, because it will erode into your personal free time, right? So I end up working on this at night at home instead of watching TV or whatever, but I enjoy doing this. But for other people, this definitely would erode into your personal time, and it's not the right answer for everyone, absolutely.
How has the history of the treatment of anal squamous cell carcinoma impacted what you're doing? Because we've been doing, you know, not even neoadjuvant, but truly just NIGRA protocol. It's just straight up chemo and radiation as your treatment of that. And many of our anal squams are HPV related as well. I guess how has that impacted what you're doing, and how is what you're finding out, how could that potentially impact my anal squam patients too?
I think it's cross-pollination of ideas. I think that's where some of the concurrent chemo radiation ideas came from for head and neck. I think that the difference between head and neck and the anal, from my talking to the radiation oncologist, that the dose that you can deliver to the head and neck, we can blast the head and neck, right? You stay out the spine, we can cook it. We can take it to levels that the bowel will just not tolerate. And I think that has been the difference so far. You know, it is interesting when you, and next time I talk to Merck or BMS, I'm going to ask them why, but what I haven't seen and what I don't understand is why they don't allow some of the anal HPV positive patients to go on to these neoadjuvant immunotherapy trials similar to GYN cervical HPV positive or head and neck HPV. I don't know why they write that group out. My suspicion is that when you're writing a clinical trial, unfortunately it comes down to dollars, right? So it's investment in and it's return out. So what you want is you want in and out fast, get an answer, and have that answer lead you to the next step so that you can prove to the FDA that this actually works so that you can start getting paid. Okay? Because everything up until the point that the FDA gives you indication is go free. And so my suspicion is that they probably think that anal cancer is a low enough incidence that they would have a hard time accruing, and so therefore, not worth it. But at some point, they're going to start tackling that area. I just don't know when. And they should, because it's almost all HPV positive.
Right, and we're still just doing, mitomycin and 5-FU.
Yeah,
We're
yeah.
doing the same thing we've been doing for decades.
Right.
And most patients do well with that, but some fall out and recur and go on to even die of it. But that's
Yeah,
why I wonder if
Get
immunomodulation wouldn't have a role.
Well,
Because
I'm
really-
scared PI on this. It's a Bristol-Myers Squibb study, but they received 2 doses of neoadjuvant anti-PD-1 for HPV positive cervical and HPV positive head and neck. And so far, the trial has seen about a 35% response rate just with the 2 doses. So there is a very strong suggestion that it would help with any HPV positive solid malignancy, and they should be doing it.
That's an interesting point. You said you have 70 grade for the head and neck.
Yep.
Our pancreatic wall is 50 to 54. They will
Exactly.
discuss that.
And that's the limitation for anywhere where you have valve. You can't crank up the radiation dose, and so I think that's really has limited some of the ability to synergize between chemotherapy and radiation. But I will tell you, on the other hand, that drives a lot of toxicity. So even though you can, doesn't mean you should, right? And so I think that yes, our patients are happy to be alive, but a lot of them do have, I wouldn't say miserable, but very different lives after their treatment. And so that's where the field's going now, is how do we back off from that very high radiation dose? How do we drop the toxicity?
Thank you.